So we're delighted tonight to have Nishad Jayasundara. I've known Nishad for many years now, actually. We met first in Boston, working collaboratively on modeling kidney health and disease in the zebrafish. And so that was the basis of our first introductions. And then Nishad, of course, has been working on this big problem of kidney disease that's of unknown etiology that spans the globe. There's an equatorial agricultural associated incidence of kidney disease that's really shocking. I think like young people are getting chronic kidney disease, which is unheard of really in different environments. So figuring this out is critical. And Nishad has done a wonderful job of using a simple genetic model, the zebrafish, to really dig into that problem and correlated that with clinical and medical records of people in different places in the world. So while we were working, I told Nishad I was thinking of coming up here for a job, and he said, take it. And so Nishad gets his finder's fee for me, apparently. And here we are, still here working together, still talking. Just sent me some data yesterday, so you'll hear about that next year. So Nishad, thanks for coming.
Thank you. Thank you so much. And I might double. Yeah, perfect. I'll keep this here. Thank you so much, and really wonderful to be here. And when Jerry and Rebecca first asked me to come for the Science Cafe, I thought, oh, I'll give a typical data talk that I usually do. And then we had a call, and then I realized here's a great opportunity for me to really learn a few things from the audience, because we have a community of folks here who are deeply engaged in science in many different ways, and what the value of science that well, at least I should say that there is incredible appreciation for value of science and what it does for society and community. So what I want to do is a slight change of title from here, and this would probably be, this is a zebrafish kidney that you're seeing, the only time you will see it in the rest of the talk. So so sorry to take the zebrafish part a little bit away from you, but what I do want to do is to talk a little bit about this broader topic of environmental health. And I think in many ways in the United States and in the world, we are at a turning point in terms of how we think about the environment and how it influences our health. So what I would like to do is to use this kidney disease example that we've been studying as a way to get you to think a little bit about what does it mean to be a conscious citizen of the environment in a way that it means that it influences our health. So let's dig in a little bit, and I want to start with a very light question to all of you. If somebody asks you this question, what are the major causes of death in society today, especially premature death? And I'd love to hear from the audience about what comes to your mind. If I ask folks in my own community back in Sri Lanka, they would say mosquitoes, because right now dengue is a big problem, and it's at the forefront of everybody's mind. So if I ask you, I'm curious to hear, does anyone want to share a thought? Yeah. Measles. Yeah? Yeah? Measles. Any other? Gun violence, yeah. Heart diseases. Anything else? Any other ideas, thoughts? Cholera, yeah. Anything else jumps to mind? So Lancet, which is, I'm sure you're aware, the journal that you're aware of, would disagree with you. So this is looking at the last 30 to 50 years of data, depending on how you look at it, and really trying to quantify what are the major causes of premature death. So there's obviously age-related death that's quite hard to quantify in terms of a direct cause. But when they looked at this data, this is from 2022, they wrote the same sort of idea in 2015, and this is an updated version of that. And what you're seeing is what they've identified as the major causes of premature death. So starting with gun violence, war and violence, about 0.6 million people a year, road traffic, infectious diseases, HIV, TB, malaria, to alcohol, BMI and obesity and atherosclerotic diseases. All of them combined is still lower than the contribution from pollution to premature deaths in our society. So 1 in 6 people, about 9 million deaths per year. And they claim this is an underestimation of this number because you can't quite always quantify how pollution might be influencing people's health. So this is, in some ways, a scary data set. But on the other hand, this is something we can do something about, right? And that's sort of what I want to sort of position the rest of the talk, is to really think through how do we, as we move forward in society for the next 50 to 100 years, how do we really think about environmental health as a discipline, but also as a key factor that would influence our own health. So when we think about the environment, there are a number of exposures that I'm sure many of you are aware. These are the sort of 5 major categories that we think about. Thanks to ChatGPT for letting me really come up with a visual like this. So air pollution is, I'm sure many of you are familiar, is a major one. Lots of industrial emissions, vehicle exhaust, wildfire smoke, which we keep hearing about now due to climate change-driven effects, and then particulate matter. There's water pollution. There's a huge problem of contaminated drinking water, which is a big problem in North Carolina at Duke University, where I'm faculty, and we've been studying contaminated river system in North Carolina from a chemical called PFAS, which I'm sure many of you are familiar with. And then there's indoor pollution, which we think a lot less about, especially in areas of the country or the world where you do spend a fair bit of time inside. So winter times in the US, in Maine rather. There is quite a bit of indoor pollution that's been identified to contribute towards many number of diseases. So thinking about the sources of that indoor pollution is really important, of course. There's contaminated food consumption, and then there's obviously consumer products that we could think about. So all of these contribute towards an incredible number of different exposures that will potentially have a health outcome and contribute towards potentially some kind of adverse health outcome. And we call understanding all of this the study of the exposome. I'm not sure if you're familiar with the term. So there's a whole field of exposure science that's emerging that's called exposomics. And this is a tool that we are using in our lab to try and characterize what the populations that we're interested in and what they're exposed to. And when we think about the sort of scope of exposomics, it's in many ways not an easy problem to tackle. So there are about 150,000 to 180,000 chemicals that are currently listed depending on which database you look at, as chemicals that are released into the environment by one way or the other. Of that 150,000, we understand the toxicity of them, about 6,000 of them. So we understand very little about what majority of these chemicals do to our bodies. There are about 75,000 different chemical mixtures and polymers that have been identified. There's about 120,000 substances that we cannot identify. So you can get a trace from your whatever the LCMS or whatever kind of analytical instruments that people are using. You'd get a trace from it, but we have no idea what that chemical actually is or where it's coming from. And then when we think about the regulation of these, there are many number of different regulatory policies for these. So there's regulatory policies that are different for industrially released chemicals versus agricultural chemicals versus the chemicals that are coating our frying pan, for example. Right? And all of this is regulated under TSCA, or Toxic Substance Control Act, that was passed in 1976. And this was around the time that I think US really led the way in thinking about how we regulate chemicals in our communities and societies to minimize health effects of those. So one of the key parts of TSCA is that it says that evidence concerning the safety for humans and the environment be provided for all new chemicals and all old chemicals with a new use. So that's a bit of a tricky sentence because it's basically saying if the old chemical was continued to be used for the old purpose, we don't actually have to know what it does. And then there is this grandfathering that happened where a number of chemicals were grandfathered in where they didn't have to provide evidence for toxicity at all. So there's there's a scope of problem in terms of the number of chemicals and the mixtures we are dealing with. There's also a regulatory bottleneck that we are also dealing with. And on top of all of that, we now have massive effects of climate change that we are seeing around us, which are in many number of ways that either lead to increase in release of these chemicals or change the way that our body responds to these chemicals. And I'm trying to understand the interaction between climate-driven effects versus pollution-driven effects, and the interaction of the two obviously is also a massive problem in front of us. So not to scare you, but I do want to say, welcome to the Anthropocene, which is the era of geology that humans have had the most significant impact on Earth's atmosphere. So if somebody from a million years from now digs through the crust of the Earth and identify all the different geological eras, today you would define it as the Anthropocene, although I think a lot of geologists still debate if this is the right way to characterize it. But I do think it's fair to say we are living at a time where environmental change is a major driver of human health. And that is, in many ways, undisputed. And I think a really important and striking example of this is this kidney disease that Ian mentioned that we've been studying. So this has been called sort of the canary in our times in terms of environmentally driven disease outcome. New England Journal of Medicine called it a sentinel disease that we should all be paying attention to, that is tied to environmental change. What's unique about this particular type of kidney disease that Ian mentioned, also mentioned, is that usually when we think about kidney diseases, it generally tend to affect as you get older. Generally, we start to see kidney diseases as you get older. But in this case, we are seeing very young people, sometimes even at the age of 35, dying from end-stage renal failure. So their kidneys have gone from perfectly healthy to completely dysfunctional within 10 to 15 years. There are no other risk factors associated with this particular form of disease. So usually, kidney diseases are found in individuals that have other risk factors, such as diabetes or hypertension. But in this case, these are perfectly young, healthy individuals who show up at the doctor and say, my body hurts, and then they'd get a series of tests done. One of them would be kidney function, and then find out their kidneys are almost failing. And in these cases, often men are impacted. So it's known as the widowmaker disease, because in a lot of the communities that are affected, primarily men die from this, and women tend to be affected a little bit less so, but there is potentially biological reasons, but also cultural reasons, which I'm happy to think through if anybody's interested. So I just want to show you the distribution of the prevalence of this disease. So when I first learned about it, was from a newspaper article that I was reading from Sri Lanka, which is where I grew up and spent the majority of my early childhood in. And I started reading about how this mysterious kidney disease is killing farm workers in one particular community. And I was starting my postdoctoral work at that time, and I started paying a little bit more attention and wrote a grant and started doing this work. Along the way, we published this paper that is currently showing this map. Is in that paper where we were finding that number of locations around the world are showing this emergence of this unexplained or unknown etiology-driven kidney disease. So Sri Lanka, India are two countries that are affected, and then a number of countries in Mesoamerica. So Mexico, Guatemala, El Salvador, Costa Rica, Panama. Number of farming communities in these countries have a high prevalence of kidney failure among farm workers, and no one really understands why. When in one community in Nicaragua, 1 in 3 men are affected. One community we worked in India called Dhanam, About 60% of the men are affected by this, and many have died from it. About a five-fold increase in number of people dying from this in El Salvador and Nicaragua over the last 20 years. And then in Sri Lanka, at a given time, you have about 20,000 people on end-stage renal failure, so basically receiving dialysis, obviously until they get if they can get a kidney transplant, they might recover, but otherwise they don't really have a lot of options in their life. So as you can imagine, has a lot of socioeconomic impacts on the communities, and a lot of people have, or researchers have tried to understand the disease and have worked on it, including our lab. And now, because of this attention to this from these few countries that were original hotspots, number of other communities have started paying attention. So now we see emerging evidence from parts of East Asia, Thailand, Cambodia, Vietnam. We have some studies in Kenya showing similar phenotypes. Other studies coming up in Nigeria, Ghana, Ethiopia, and also in the United States. There are 3 or 4 described populations that are showing very similar disease outcomes, especially in Texas, which is one of the first places to show that there are farm working communities who are affected by this. And then we have work now going on in North Carolina, again showing that there are farm workers affected by this. A lot of the work we've done is based out of Sri Lanka. Obviously, because I grew up there, I feel certain affinity to go back and do this kind of work. But also Sri Lanka is a central location, partly because of a really highly organized public health system. That means a lot of documentation, so you can get the types of data that you need to look at long-term disease trends. There's a huge, incredible public awareness, a lot of efforts by the government to make sure that people are aware of this disease, and because of that, number of resources allocated towards understanding the disease. And if you look at the disease distribution, it's not the whole country that's affected. So Sri Lanka is it's kind of the size of Maine, and if you look at the regions that are affected, parts that are here in white where we don't see the disease incidence, those are nice and cool. Although there is farming, we don't really see the disease impacting farm workers. So they're some of the milder climates in the country. And then you have these incredibly hot and dry regions of the country that are colored in red and pink that are what we call the endemic hotspots. And in dark brown, you're seeing one of the biggest, the most affected areas where 5 to 20% of the population is affected by this disease. And we've been doing work all around these communities, collecting various kinds of samples. And this is what one of the communities look like. So when you go there, you don't quite think that 5 to 10% of the people or 20% of the people are affected by a disease that's going to be a terminal disease. It's a fairly normal community life that people are living there, and these are some of the field pictures. The 2 folks at the end, this is Dr. Chamal in green. He's a physician from one of the affected communities. His mom actually died from this disease. So one of the fewer examples of women being affected. And then Mr. Saman, another community member who's instrumental to all the work that we've done. He's holding my son on his shoulder. So I tell him not only our field work is dependent on him, but also my children's life is dependent on him. So he's been an incredible member of our work that's really helped us to do the type of research that we want to do. I also want to highlight Mangala De Silva. He's a professor at one of the universities in Sri Lanka, University of Pune, who's also been the main sort of supporter and a leader of this work in the country. And then we set up our own nonprofit to lead the work in Sri Lanka in a way that we can have policy discussions with the government based on what we find, so we don't put necessarily Duke on the spot when we go to the government with the data that we are finding. So this is sort of the body of the work that also the foundation of our research efforts that's helping us to ask questions around what's causing this disease. So I'm curious to hear if you hear about this on the news or in some other format, what would you try to do to understand the drivers of a disease like this? So I'm not sure if anybody is willing to give me some ideas, but I would love to hear how you would go about thinking about this if you have any thoughts. So there are communities in the US affected. So.
And I would look at common dietary habits.
Yeah.
I test the well water.
Test the well water, yes. Yeah. Any other thoughts on?
Check the air.
Check the air, yeah. Air monitoring.
I'd look at temperature and hydration.
Yeah, exactly, temperature.
Going back to the toxicity, like if there's any shared chemical that are all.
Right. So what I'm generally hearing is there's heat, so temperature and hydration, and then air, food, water contamination. I saw another hand.
Controlled studies. We analyze the kidney biopsies and toxicology, terms identifying the genetic toxins.
Yeah.
And affected patients versus
Unaffected patients. Yeah.
Well, what kind of pesticides have they used? These toxins have compared to other places in the area that aren't hotspots.
Right. Right. So look at the disease affected community members versus unaffected, and then think about the differences in chemical usage or pesticide usage, and look at water sources, air sources. Any other ideas or thoughts? Yeah.
I just wonder what kind of healthcare everyone's getting and how well the disease is being monitored. This would be a big silent population that you could miss.
Right. So better community level population health monitoring is needed. So who's got the budget for this now? That was the real question at the end. No, but I think incredibly good points, and that's where we are headed, and that's what in many cases we are doing. But based on your answers, I'm sure it's fairly clear that this is a vast landscape to really think about. So we want to monitor patients, biopsies, blood and urine samples from affected individuals versus unaffected individuals. Obviously, one snapshot is not going to be sufficient, so we want to do a controlled study and do a long-term longitudinal monitoring. We want to measure water to make sure that there is nothing in the water samples that we could collect from these communities that would point to a potential causative agent. Air monitoring, we want to look at food. We would want to look at what's in the indoor environment. And then obviously temperature as a variable that might be modifying either the effect of the chemicals or temperature directly could be having an effect. So that's basically our research program, is to try and look at these different variables. So what my lab has set up to do is to, through many collaborations, is to quantify the environment as best as we can. So look at occupational and agricultural exposures through various cohort studies, environmental monitoring, and then do exposure assessments, both in human samples as well as environmental samples. And then also quantify disease pathology using biospecimens we can collect from patients and healthy individuals. I just shared some of our latest data with Ian yesterday that he mentioned, where we have a collaboration with the local hospital that provides kidney care to one of the reddest spots that you saw on the map. So we have kidney biopsies coming from patients who are at stage 3 and stage 4 kidney failure, and also kidney biopsies from other kidney disease types, so diabetic kidney disease or diabetic nephropathy, versus healthy individuals. Obviously, there's an ethical concern, so we don't take biopsies from healthy individuals, and that's a big limitation obviously for our studies. And then a factor that I think we've really not thought a lot about in all of this is that let's say we could measure the environment as best as we can. But if you find 1 or 100 or 1,000 chemicals in the environment, how do we know it's actually going to affect the kidney? Which database should we look at? And is there enough data in our existing databases to really point towards nephrotoxicity? And at what level would they be causing toxicity in this population? Because they may be exposed to not just one, but probably 20 other things. So that's really where the zebrafish model has become really important for us. And again, Ian has helped us develop several tools and number of other researchers around the world that help us screen for these chemicals in a very short window of time. And we can test multiple different chemical combinations to see which one of these chemical constituents in a drinking water sample, for example, might be affecting kidney function. So one thing we've done, for example, is to go into these affected communities, collect hundreds of water samples, bring them back to the lab to Duke, and then expose developing zebrafish embryos to this and look to see which of the water samples are leading to potential adverse kidney effect based on the markers that we are looking at. So several different gene expression markers as well as functional markers that we can look at. So the zebrafish model has really helped us to bridge the information we get from the environment and try to link that with the molecular pathology we are seeing in patient samples to see, can we detect the same changes in our fish models? Obviously, this is incomplete in many ways, right? But this is one attempt towards trying to better understand how environmental change might be driving this potential disease outcome. And I just want to show a couple of slides on environmental measurement and also one of the data points that we have. So when we heard that, oh, you can measure water, air, food, measure temperature, I'm sure you can imagine it's incredibly complicated. So one of the things we do to measure chemical exposures in the indoor environment and also outdoor environment through aerosolized exposures is we put in these silicone wristbands in our farm workers. So when they're spraying chemicals or when they're walking around in their village, they're continuing to gather the different types of chemicals that might be in that environment. And we can take a little piece of that little wristband that they're wearing and then bring them back to the lab and do several different chemical analyses to figure out what are some of the chemicals that these individuals might be exposed to. We actually have developed now assays to be able to extract those chemicals from the wristband and do zebrafish exposure studies. So we can not only figure out what chemicals are in the wristband, but also are there chemicals in the wristband that will affect kidneys in a developing zebrafish. The other thing we could do is to go to drinking water sources, well water sampling that Jane mentioned, and collect a ton of these samples, bring them back to the lab, and again, do the same types of studies. And then we also do heat exposure monitoring. So those are two postdocs in the lab who's done a lot of work to try and better understand and develop tools to better monitor heat exposure among these farm workers. One of the, I'm sure you hear on the news, so it is the heat index is higher today, or temperatures are over 100 degrees today. What we don't quite understand is what do those temperatures actually mean in terms of kidney function? And that's just emerging body of data right now, including the work that we are doing. So one area we are working on is to do personalized heat monitoring. So we have these little instruments that they wear as they work in the field. Each one of those little things costs about $1,500. So we follow our farmers to every day to make sure that they don't lose it. And they wear it for 7 days while they work, but also while they sleep. And we can get a sense of their core body temperature. And we also have these monitoring stations set up in their farmland where we can also get environmental temperature. And then there's remotely sent satellite data that you can get that also give us a sense of the area-wide temperature data. So using all of that, there's another postdoc who's not depicted here, who's in the lab just starting to really put all of those data sets together and look for associations with the markers of kidney injury that we measure in these farm workers. There is already clear data showing that temperature increases can affect kidney function, but we are trying to better understand how does that affect a farm worker in a field where they're working 6 to 8 hours in the hot sun. And what does the recovery look like? And one of the things we are finding is that farmers who get a good night of sleep in a cooler environment actually recover from that kidney injury they experience in the farm during the day, actually are better off in the long run after 6 months, for example, as opposed to farmers who don't get a good night of sleep in a cooler environment. The other sort of interesting data point that I want to share is the children who live in these communities. So one of the aspects we learned from the zebrafish work is that these chemicals in the drinking water can affect kidney development. So we wanted to do a screening across these disease-affected communities. So we have 3,500 children enrolled in a cohort study with us where we continuously monitor their kidney function based on urine, which is not the best way to measure kidney function clinically, but we also don't want to collect blood from a child over and over again. So we only do urine-based analysis, but what we find is that if you're a child living in, this is the only data slide I think I'll show, if you're a child living in a CKDU affected or kidney disease affected village, we have a much higher probability of finding a child with the increased marker of this one particular marker called kidney injury molecule 1, which is indicative of proximal tubular injury in the kidney. And we find that kids in the affected areas are more likely to show this induction compared to kids who live outside of these villages. And another nephrology fellow, sorry, I skipped that, that I've been working with, Anna Strasma, who's also leading some of the work in North Carolina with us. She's done a meta-analysis across the world and found that if you are a child living close to an agricultural community, you're much more likely to have elevated kidney injury compared to a child who live outside of that agricultural community. So not only we are seeing that adults are being affected from whatever the exposures are in these areas, but potentially disease incidence might be tied back to some type of early life injury that might be taking place. So we are continuing to do that work in Sri Lanka. These are all of our cohorts. I just wanted to show you that we are going all around the country, visiting schools and working with children to give us their biological samples and try and monitor what are the potential changes that are taking place in their kidney. So just to summarize everything we found before I move on to the last bit of my slides, is that we found that exposure to heat stress and dehydration are directly linked with induction of these kidney injury markers. We've also found that there are a number of contaminants in the drinking water that is linked to increased kidney injury. Specifically, we find heavy metal mixtures, even at very, very low levels below what's considered safe for us to drink. So arsenic, cadmium, and lead, and also vanadium are four elements that keep coming up. Glyphosate, which is a pesticide that's used all around the world, is the most used pesticide. We keep finding an association with glyphosate and kidney injury. We also find fluoride to be associated with kidney injury, but the associations are not as strong as some of the others. And also consumption of hard water, so water that contains higher levels of minerals. And the other main, most important point that we keep finding is, as I mentioned, is that there's potentially an early childhood or early developmental exposure that might be preceding this disease incidence that we should be paying attention to. So why are kidneys so sensitive to the environment? And just to show you that, I wanted to first start with this, but I thought we all know what a kidney does. So kidneys are supposed to be filtering out all the xenobiotics and waste that are produced in our body. So in a given day, about 7 bathtubs full of blood comes to our kidney and filters out, where we excrete about 2 wine bottles full of water at the end. What that means is that kidneys are experiencing all kinds of contaminants that are circulating through our body and are being concentrated at the sites of filtration, so at the sites of these tubules. So kidneys are sensitive because potentially the dose in which a kidney tissue is exposed to a chemical is much higher compared to the other organs in the body. The other part to pay attention is when it comes to heat, the way that we try to dissipate heat from our body, where we vasodilate and we reduce blood flow to some of our peripheral organs, including the kidney. And by doing so, there is potential hypoxia or lack of oxygenation into our kidney tubules. And that is being shown to also lead to acute kidney injury. And there's data showing that even as there's data showing that about 50% of the people who've been reported to have acute kidney injury early in life are much more likely to develop chronic kidney disease later in life. So even a mild acute kidney injury episode could actually increase your susceptibility to some of these other toxins, or just on its own can lead to chronic disease stages. So with all of this work, what has come together for us is that there are a number of different environmental variables that are directly targeting kidney health. There are occupational exposures, number of other environmental exposures, heat and climate-related stresses, as well as lifestyle factors. But when you look at how we've studied the kidney over the last century, let's say, a lot of the attention has gone into really understanding the biological susceptibility to diseases. So I think there's a significant need for us to think more about how this environment might be either interacting with some of these biological susceptibilities or on its own driving kidney disease outcomes. And I think this is my last couple of slides. One reason why we should start paying attention is we are pretty much born with the nephrons, or the functional units of a kidney. And every time you have an insult to those functional units, unlike zebrafish who can regenerate, humans don't necessarily regenerate that functional unit. So the more damage a kidney might experience through time, the more likely that that child or that young individual may experience a chronic disease outcome later in life. So when we think about medical interventions today, we think a lot about interventions at the stages where we can actually measure a significant functional change in our kidney. But I think there's a greater need to really think about how do we intervene much early on. And to do that, we need to better understand the biomarkers of some of those early injuries. And that's something I hope that the zebrafish work that we're doing in collaboration can really help us understand. But one thing I want to point out is that the very early kidney development actually happens within the first 5 weeks. So even before one might know they're pregnant, they might be developing a fetus where the first forms of the kidney is being developed or are being developed, and might be susceptible to some of these contaminants that we just showed. And this is important because obviously more than 800 million people suffer from kidney diseases. 1.2 million people die annually from kidney diseases in the US, projected to be the fifth leading cause of death in the world by 2040. But also really strikingly, about 20 to 40% of the Medicare budget is spent on providing kidney care in the United States. So again, we paid a lot of attention to thinking about biological susceptibilities, but I think there's a really significant need to think about how environment might be also contributing to this problem, and think deeper about which communities are affected and how do we make sure we protect those communities from those environmental variables. So I'll leave you with this very extravagant, exaggerated, I should say, Time magazine article that said chronic kidney disease is poised to become the black lung of climate change. I'm not sure if I would go that far, but I do think there is a need to really think about this disease, the role of environmental change in the context of this disease. And I think one other question that I want to ask is, yes, we did all this work and we found chemicals are bad for the kidney, we found heat is bad for the kidney, but we really don't take a lot of action right away for these types of environmental variables that can have disease outcomes. It's been incredibly challenging in Sri Lanka to get the government to be, for example, interested in providing clean water for all of the communities that are affected. They do to a large extent, but not enough. But also, there's a lot of roadblocks when we talk about environmental variables driving disease outcomes. Part of that is also the scientific framework that we have in front of us is not always adequate for rapid policy making. So when it's clear from the work that we've done, there are long-term effects, there's delayed effects of these exposures, multiple exposures, differential vulnerabilities, interactions with other stresses, all of that contribute to a poorer understanding of a disease outcome because we can't quite link it right back to a cause, right? But we also live in I think, a time where we like to invest a lot in drug development and to cure a disease more than prevent the disease. So there's, I think, a need to really think deeply about where do we invest our resources in when it comes to creating or living in a healthy environment. And who is responsible for doing that? Who cares? I think the EPA has a big role, the National Toxicology Program, which recently shut down. The HHS is another responsible party. And then NIEHS, National Institutes of Environmental Health. All of these agencies are critical in ensuring we understand how chemicals affect our health or how environment influences our health. But all of this actually in many ways ties back to this movement started by Rachel Carson many years ago. I'm not sure if you've read the book Silent Spring. If you haven't, I highly encourage you to. And US really led the way in the world in creating these agencies and institutes to make sure communities are protected. And she played an incredible role in getting the nation excited about creating these types of agencies to protect our health. But when you look at the budgets allocated for agencies like NIEHS, you could see that only about 2% of the total NIH budget goes to environmental health, although in my opinion, as that Lancet article also showed, environment seems to play a much bigger role in how healthy an individual is in their community. And when you correct for inflation, you actually see that NIEHS budget has actually decreased over time. So while I do want more money for this work, I also wonder, do we really need to fill all the data gaps before we take action? And how much of a risk do we put on the future generations while we better understand how a given chemical actually affects somebody's health? So with that, I'll end the presentation, and I just want to thank an incredible amount of collaborators and students and community members and also all the funding agencies that supported this work, and happy to take questions or anything. Yeah.
I'm curious what the reaction has been. Have you sort of interacted with the agricultural companies or the manufacturers? What is their take on this data?
Yeah. So Monsanto, which produced glyphosate, now no longer is in business. They have been present in many of the meetings that scientific meetings that have had this data presented. So there's always been pushback on the role of glyphosate. In Sri Lanka, the government actually paid attention to the data that were coming from our group and others. So they banned the use of glyphosate, and that led to a massive backlash from the farming community because they want the pesticide to be around to make sure they can grow enough food. So farmers themselves were more willing to take the risk of using glyphosate while they feed their family and then hope that the risk of kidney disease is only 20%. So they were pushing back against the government, so they reversed the ban, but now it's banned again. I think the South American communities that are affected by this, they really point to heat as the only driver of this disease. We don't necessarily see that in Sri Lanka. And data coming from North Carolina show us that heat is a major player, but not the only variable. So North Carolina farm worker data is suggesting time spent outdoor working under heat can lead to kidney injury, but also agrochemical exposure is directly associated with this. So it seems maybe there's a potential interaction between the two. Yeah. Steve, yeah.
Thanks, Nishad. I thought that was terrific, if not scary. But the heat and dehydration go together as they did in your circle.
Right.
And so I have two questions about that. Number one, all of the places that have the CKD are hot and dry.
Right. Yeah.
So heat minimizing heat causes thirst and for a lot of reasons, and that requires people to drink a lot. And so if the water is polluted, that is aggravating the problem. That's the first thing. And then the second thing is how do you understand the risks or the role of dehydration itself, especially if you're using a laboratory animal that's a fish in fresh water?
That's an Ian question. Great points. And so within Sri Lanka, we have studies in non-farm working communities, but are extremely hot. And we work with salt pan workers. So these are basically areas where people let seawater come in, and then they bound it. They put a little wall around it and keep the seawater in these little flats, and then let the water evaporate, and that creates a layer of salt, and then you have these workers go and break the salt off. So they're working under no shade, incredibly hot and dry conditions without drinking any water. And when you look at EGFR or filtration rates, they have the lowest filtration rates compared to anyone, but they don't have chronic disease outcome. So what we see is that people who work in farmlands have a less of a reduction in kidney function through the work environment or to their working day, but they end up showing this chronic kidney disease phenotype. Whereas people who are in the salt pan areas, we find very little chronic disease outcome rates. So there is some disconnect between the acute change in kidney function that's happening from dehydration and heat versus how much it's going to contribute towards a chronic disease outcome. So it is possible, as you said, that perhaps you are drinking contaminated water, and that's actually what might be leading to the initiation of disease phenotype that ends up leading to a chronic outcome. And the studies with the fish is really obviously not to think about dehydration. We are using that primarily as a toxicity screening system to better understand which chemicals should we be paying attention to. And Ian is part of this work. We have a grant that's almost completed now where we are comparing fish data with mouse data and organoid data, where we can ask some of these same questions, but obviously not drowned in water. So, yeah. Yes.
John, I was curious about the children, the screening study.
Yeah.
And you mentioned that when they're closer to it, if they belong to farming families, there's high susceptibility. I've been wondering about effect modification in terms of gender.
Is that, sorry?
I've been just thinking about effect modification, just
Yeah.
in between two genders. I understand in adults, the males are more because of their in low middle income countries because of the occupation they're outside. But I'm just thinking about kids, and do you see differences between boys and girls?
Yeah, great question. We do see, we actually see that young women or young children, female children in the communities show a higher induction of kidney injury or KIM1 molecule, but lower disease outcome at the chronic disease stage. So There is some work coming actually from someone at Duke who's shown that female mice have a better antioxidant defense system in their kidneys, and it's possible they're more protective against the kidney injury that's taking place early on in their life. We also see that girls tend to drink less water because they have to go to the toilet and the toilets aren't good. So it's also possible there's a more dehydration-driven injury that we are measuring that's actually not contributing towards the disease outcome later on in life. Also, women work less in the farm land and men do most of the work. So it's possible that the second hit or the early life risk or early life injury is occurring through contaminated drinking water consumption and potentially heat injury, but then they go on to become farm workers and their kidneys are already susceptible to whatever the chemicals that are there. With the North Carolina farm worker populations, we don't see kids being affected. We haven't looked at it, but we don't, none of the clinicians would point to anybody showing up at the hospital with kidney injury at a young age. But then on the other hand, we don't monitor kidney function in kids in the US. We rarely do. So it's possible we might be missing that as well. Yeah.
Is there a genetic component to this?
Yeah, there's some work that showed there is no genetic component, but I don't think that was exhaustive enough to really say there's no genetic component. The fact that the disease is emerging across very different communities around the world with very different genetic backgrounds suggests to me that even if there is a genetic component, there is a clear environmental driver that is triggering that particular genotype to be somehow maladaptive. So it is possible because they are living in a tropical, in the tropics. And we do know that, for example, there are these communities in Turkana in Kenya, which is one of the hottest places on Earth. And they have these solute carrier proteins that function differently and have a genetic mutation that enables them to retain water better. So it is possible that communities that have evolved around the tropics do have either acclimation-induced or genetic changes that enable them to be healthier in the hot tropics, and maybe that contributes towards this disease phenotype. There's a nice paper in PNAS recently from I'm blanking on his name, showing that there is a particular phenotype that are commonly found among South American farm workers, genetic phenotype, and they captured that in a mouse model, and they showed that if you have that phenotype, you are more likely to develop a kidney injury from heat exposure than if you're not. They tried to initially attribute that to this genetic bottleneck that formed while communities crossed the Bering Sea. So you had to survive the cold environment. So the reverse phenotype of being in the tropics, which allowed them to survive the cold environment, but now that it's working against them in the tropics. And so somebody gave a talk on that, but then in the paper, they didn't mention that part. So I'm not sure if that got sort of removed by the reviewers, but I thought that was a fascinating way to think about it. However, there isn't really clear evidence to suggest there's a genetic component. We also don't see family. We see families, but it seems to be a lot more linked to what they're exposed to more than one brother who moved away doesn't really show a disease phenotype from the village, for example. Yeah, that's right.
Can you talk about the futures research? I'm curious if regulators need like a certain amount of research before they take action, before they take you seriously. And also, what has your experience been like working with regulators for toxic chemicals in different countries?
Yeah, no, that's a great question because I think it depends on, in my experience, it seems to depend on your intentions going into the policy making, right? So if the intention is to protect community health, they're willing to work with very little data to push policies towards protecting communities. If the intention is driven by, let's say, lobbying from a company or lobbying from the farmers who say we want the chemicals to be around, then it doesn't seem to matter how much data you show. then the policymakers are always asking for more data, more conclusive data, more concrete data. Right? So it's been a really interesting space to be in terms of who wants more data, and it really seems to come down to the intention even before they see the data.
Nishad, I mean, are there things we could do differently that would help policymakers to understand? I mean, you had a slide that sort of outlined some of those key things. I'm just curious what your takeaway is from your experience there.
Yeah, I mean, obviously, I get paid to do this work, right? So to me, it's very clear that I think before we try to generate more and more data, I think we can do, especially what Europe has done, where the onus is on the chemical companies, not on the consumer. And there could be a lot better set of rules in place for entities that are producing these chemicals to show that there are no adverse effects for the environment or to human health, but that doesn't quite exist here. So I think that would be a big flip in our policy positioning. So that's something I would like to see first, is before we study the chemicals, but to just have the mindset of we have to have better data before we release a chemical out into the environment. And I think that would be a great starting point. But even for chemicals that we know so much about, for example, arsenic that Jane studies, and we're still finding that there is no safe level of arsenic, right? So then there's also this premise of obviously we'll never have no chemicals in our environment. For example, we recently came across this data set that showed the sheetrock in the buildings have these fungicides that are especially in the warmer climates to reduce mold growth, right? And that is found in people's blood if you live in a house that has those fungicides. So if you drive a brand new car, the new car smell you get, that's also associated with a whole bunch of volatilizing chemicals. So if you take a blood sample, you could say, oh, you've been driving a brand new car last week, right? So there's dryer sheets, for example, carry a lot of these chemicals. Different types of chemicals. Your sunscreen carry a lot of these types of chemicals. So there's really no escape with the way that we've structured our lives currently from these. But I think there are definitely ways to reduce what's in our water and what's in our food and what's in our air.
Are there any data in China about this where they have the better definition of environmental exposure?
Yeah, great question. And we recently put forward a paper with databases available around the world that have been produced, that are nationally sponsored to inform risk. And Korea and Japan and Hong Kong and Taiwan, they've all done taken a massive effort towards documenting. We couldn't find much coming from China, and we also couldn't find much coming in terms of publicly accessible data. We also couldn't find much at the regulatory level that would tell us about what some of these chemicals mean. Another challenge is that once some of these compounds become, the patent is expired. So for example, glyphosate, the patent is expired, so anybody can make it. So we only know the toxicity of the active ingredient. It could go into a formulation that stabilizes the chemical and also can have other ingredients in it that might also serve as potential pesticides. And depending on which country is producing, you are still buying a can of glyphosate, but if you're buying it from a country that has some of the regulations that are different from what's in Europe or in the US, you might be exposed to a different set of chemicals. So there has to also be not only a national effort to your question, I think there's some level of global recognition that has to come to really start thinking about what we are exposed to.
There are actually two questions from the online attendees. One is, what makes nephrosis such a difficult disease to identify a cure for? And the second is kidney disease also a problem through the use of everyday drugs like ibuprofen?
Second, yes, NSAIDs are fairly detrimental, but I think unless you're already at high risk of getting kidney disease, I wouldn't worry about it too much. There is evidence on, for example, athletes, they're told if they've worked really, if they practice really hard in the hot sun, they should try and avoid getting an NSAID that day, and don't drink alcohol and don't take too much medication to alleviate pain. Why is it hard to cure? I mean, I don't know. I think I would go back towards Ian and Herman on this one. Why is it so hard to cure kidney diseases? I think once our nephrons start to degrade, we haven't really found a way to reverse it.
Kidney disease goes undetected.
Right.
I think then during that phase, there's progression, continued insult, and chronic inflammation, which will result in essentially a big scar where you had a kidney. And so the real challenge is restoring kidney function, and that's where regenerative biology comes in. You can think about how fish regenerate their kidneys and how we might take some clues from that, but it's really about preventing initial injury, I think, and that's why your work with children is so important.
Right, until we find some way to reverse some of this, right? No, I think the work from children from these agricultural communities are showing that there is a risk to childhood kidney health. So there could be much better monitoring in the US, for example, especially in at-risk communities, community level screening that can happen that would hopefully prevent later life disease outcomes. But every time we this particular work, every time we send it for grant to NIH for review, we get back, is this a risk to the US populations? And it's so until we know that, perhaps we might not know more. Yeah. Yes.
Do you think that the ones in the southern part of the US that you're talking about the US, do you have enough information that it looks like it's moving because of climate change? Like, enough historical information in a way?
Yeah, wonderful question. And I kind of, there's a whole group of researchers around this work that really push this to be, like that Time magazine article, to be the climate disease. And that's really linked to climate change. I don't think there's strong enough data to really show the 30 to 35 year change in temperature patterns that we've seen around the world really explains an increase in disease incidence. So it's very clear that heat poses a risk, and it's very clear that heat injury can increase your susceptibility to toxins. But I don't think there's enough data to really show that climate change is what's driving this. But that's part of the work that this new postdoc in the lab is trying to map. Just looking at electronic health record data around counties at the county level in North Carolina and South Carolina, and see can they detect a pattern where there's been increasing admissions rates related to high frequency of heat events. Have there been changes in kidney function in individuals who already were diagnosed with kidney disease that links back to heat wave events? But again, the link back to a 30-year data set is incredibly challenging. And I think that's the big sort of the biggest issue for this field is how do you link long-term changes to a disease outcome in an individual.
Nishad, thank you so much. This was wonderful. Really exciting work. I'm not sure we mentioned it at the outset, but for those of you that don't know, Nishad graduated from College of the Atlantic, and he was a student here at the MDI Biolab as part of a statewide research training program that we offer. So all of MDI takes full credit for your amazing work, and we are very, very proud of you. So thank you so much for being here. Really appreciate it.
Thank you, everybody. Bye.
And this is actually our last scheduled Science Cafe for the summer season, although I hear rumors that we might have an art and science lecture at some point this fall. So we will keep you posted, maybe in late September or so. So stay tuned for that. But also we'll be kicking off our winter season in early 2027. So if there are topics that you'd like to learn more about, please let us know. We'd be more than happy to think about who we might know that could speak to those issues. So thank you all so much for your support of our science cafes. We're grateful to our sponsor, Cross Insurance, for continuing to make these free and open to the public. And so I'm sure Nishad will stick around for a few minutes if you have some additional questions. Enjoy some refreshments, and thank you so much for being here this evening.